Hello, everyone. Thank you for your interest in therapeutic drug monitoring of beta-lactams. My name is Mohammad H Alshaer. I'm a research assistant professor at the Infectious Disease Pharmacokinetics Lab here at the University of Florida, College of Pharmacy and Emerging Pathogens Institute. Because of the large time difference, I decided to go with a pre-recorded session. So today, we're going to discuss briefly the changes that ICU patients might have that affect the pharmacokinetics of antimicrobials and go over some of the PK and PKPD parameters of the beta-lactams in preclinical studies and some of the major clinical efficacy data and toxicity data on beta-lactams in the clinical studies. And I'm going to also go over some of the US and our lab experience with the beta-lactam TDM. So the number of sepsis cases is increasing, and the associated mortality is high in these patients. And there are some physiological changes that affect the PK of the drugs, including augmented renal clearance, altered protein binding, which will affect the unbound fraction of the drug, changes in the volume distribution and organ dysfunction, which may affect also the clearance of the drug. And current regimens, as we will see later, may fail to achieve the PKPD target, especially early in therapy. And in general, in ICU patients, even you may see higher MICs compared to other units. So the importance of early and appropriate antimicrobial therapy will help decline the mortality rate in these septic patients. This figure just shows some of the changes that ICU patients may have. For example, they may have increased cardiac output, which may affect the clearance or increase the clearance, which will affect the drug exposure that will go down. There may be some third spacing affecting the volume distribution, increasing the volume distribution, and there will be a decline in plasma concentration. Patients may have renal or hepatic dysfunction, or might be both, which will affect the clearance of the drug, and more drug will be accumulating. Or the patients might be on renal replacement therapy or ECMO therapy. This may affect if the volume distribution or the clearance and concentration may go up or go down. So whenever we are talking about beta-lactams, we're talking about time-dependent killing agents. So in this concentration versus time profile, it shows the drug concentration going up and then eliminated. So, and we have the MIC, so the time above MIC, that is the PKPD parameter for optimal kill with beta-lactams. So, and it is represented as a percentage of the dosing interval of the antimicrobial. So in this case, for example, the time the concentration was above the MIC is about 3 hours. And if there's Q6 hours regimen, so it's going to be like 50% time above MIC for this patient. And as I said, this has been proven preclinically that beta-lactams are time-dependent killing agents. As you can see, the change in the colony-forming units and on the y-axis, and the x-axis is the PKPD parameters, and the best describer of this relationship was the time above the MIC. And if we're looking at different targets, so that is maximal kill target versus a resistance suppression target. On this figure, you can see on the y-axis the change on colony-forming units, and on the x-axis is the antibiotic concentration. As you transition from low to higher concentration exceeding the MIC, you can see the gray line drop, which is the drop in the susceptible bacterial subpopulation. But as you cross that MIC, you will see also an increase in the resistant bacterial subpopulation, which will need much higher concentration to kill these resistant organisms. So some of the data summarizing the PKPD parameters for clinical efficacy and resistance suppression. So we have for penicillins, for clinical efficacy, more than 50% time above MIC, cephalosporins more than 70%, whereas for carbapenems more than 40%. For resistance suppression, it's kind of like C-min to MIC more than 4 is generally the best PK/PD parameter associated with the resistance suppression. And this data had been proven to preclinically extensively, but not very clinical studies looking at this PK/PD parameters in patients. So in addition to the MIC, whatever is affecting the threshold for suppression of resistance, there's bacterial species, and inoculum effect also may affect this threshold. But in general, as I mentioned, C-min to MIC 4 or more is needed to suppress resistance based on mainly preclinical studies. Another layer of complexity will be what is the unbound fraction of the drug whenever you're doing TDM. So if you're looking to the left side, you can see that is in blood that is unbound drug, which is interacting with the protein. So there will be some protein binding, and only unbound antibiotic will be able to cross the blood capillary wall, going to the interstitial fluid, and also unbound antibiotic might be interacting with the proteins over there, and there might be some protein binding. And again, the unbound fraction of the drug will be the only one going to the through the cell membrane to interact with intracellular site, target site, or it might be like binding to the cell membrane. So if we're measuring total concentration in the blood, and you can see to the right the peak, it might be different than the actual unbound antibiotic acting at the site of infection. So just to keep that in mind. And now in the literature, there is enough evidence to show that there are like variations in the protein binding between patients. Whatever published values might not be the same for your patient. So you can see to the right that the orange circle is whatever published values compared to, you can see the wide range of protein binding or the unbound fraction for these beta-lactams. In addition to the protein binding, in general, the PK and PK/PD parameters, there is variability in ICU patients, as we mentioned earlier, because of these changes the patients might have. So these are 3 different studies reporting the PK/PD parameters for beta-lactams, and you can see the wide range of PK/PD parameters achieved. If you're looking at below 1 MIC, you may consider that therapy failed in these patients, or there is a high probability of therapeutic fail in these patients. Whereas for those above 4 times the MIC, the research is still going on about the toxicity associated with these beta-lactams because of the high exposure. So looking at the clinical efficacy of beta-lactams, so DALI was the biggest prospective PK study in ICU patients where TDM was done. In patients who had beta-lactams, it was more than 300 patients, and the sampling was mid-interval and at the end of the dosing interval, which was 50% and 100% time above MIC assessment. And positive clinical outcome was associated to be with increasing time above MIC of 50%, 100% time above MIC while adjusting for Apache score. And again, this is shown also again with blood infections. Some other studies try to correlate cefepime trough concentration with the neurotoxicity. Both of these studies found significant association between trough and neurotoxicity. As the trough goes up, the probability of neurotoxicity goes up, but still there is some research ongoing on other PK parameters, including Cmax and AUC. So the idea of therapeutic drug monitoring is not just giving a single dose for all the patients, because every patient is unique. Because if you give one dose for everyone, you're ignoring the inter-individual variability, which was proven in the previous studies I presented. So therapeutic drug monitoring aims to um, finding an optimal treatment for each patient by targeting a therapeutic range in the blood. And once that therapeutic range is chosen, it should be targeted with the maximum precision. In addition to doing TDM, also the timing of TDM is important. Several studies conducted in ICU patients and sampled early during therapy with beta-lactams, they found that the optimal PK/PD target attainment was poor in these patients, especially early in therapy. And there's one study correlating delay in TDM was an independent predictor of poor outcomes while controlling for time above MIC. So in the US, in general, therapeutic drug monitoring in the US for the beta-lactams is not very common. A few hospitals, like 3 to 5 hospitals, started recently developing beta-lactam assays to be used for clinical care, which is different than research, and the requirements is different. Our lab, which is the infectious disease pharmacokinetics lab, is the leading lab in the area of antimicrobial TDM in the US. So our lab is CLIA licensed and CAP certified, led by Dr. Peliquin. We have 5 LCMS instruments, and the way we run beta-lactams assay, we do total concentration, and we run beta-lactams daily, Monday through Friday. We have 11 beta-lactams: amoxicillin, ampicillin, aztreonam, cefazolin, cefepime, ceftriaxone, imipenem, oxacillin, nafcillin, meropenem, and piperacillin. The top 5 we do for 5 days a week, and the rest won't be PRN. We usually recommend sampling of peak and trough for patients and for all sick patients, and as early as possible. And per our PNT approved protocol for physicians and pharmacists can order TDM samples. And the importance of having peak and trough is that you'll be able to calculate the PK parameters, including KE, volume distribution, Cmax, trough concentration in case the second sample was not a true trough, and AUC, time above MIC. So all of that can be calculated using 2 points. And over here, we use a simple PK sheet to calculate these PK parameters, as I showed earlier in the figure. And calculate the next best dose to hit PK/PD target of interest for each patient. Sometimes we use Bayesian software using population PK models that we develop, also to calculate these parameters for these patients, but still this is not fully implemented. So at the end, I would like to thank our team, the IDPL team, stewardship team, ICU team, and microbiology lab, and all the data team. Without them, this work would not have been possible. And if you have any questions, please feel free to email me, and I'll be more than happy to respond. Thank you very much.